about my oh, hi there, yeah. I'm so excited to be talking today about my paper, or our paper rather, a flaw in using pre-trained pLMs in protein-protein interaction inference models. You could read it now in Nature Machine Intelligence. We're really excited to for you all to see it. I'm Joseph Szymborski, by the way. I'm a postdoctoral researcher at McGill University in Amina Mad's lab in the electrical and computer engineering department, as well as Mila, the Quebec AI Institute. And it's my honor today to be speaking to you all here. So let's begin by considering two proteins. Here we have ACE2, a protein expressed at the membrane of human cells, which, among other things, helps regulate blood pressure. The second is a spike protein from the SARS-CoV-2 virus that's the cause of COVID-19. When the SARS-CoV-2 spike protein interacts with ACE2, it triggers a cascade of reactions, which allows for the SARS-CoV-2 virus to enter human cells. This is an example of a protein-protein interaction, or a PPI. And PPIs underpin critical physiological and biochemical functions. And as illustrated by this example, they could be great. They could be of great medical and physiological, and in this case, socioeconomical importance. So it follows then that it would be super useful if we had some kind of algorithm or function to infer whether or not these proteins interact. Traditionally, we run so-called wet lab experiments to confirm an interaction, but those are expensive, and in this case, or the case of the example before of SARS-CoV-2 and ACE2, it can be very dangerous and risky. So I'm not the first person to come up with this idea that it would be useful to have a function to do these things. People have been working on this since at least 1996. At first, researchers employed non-machine learning methods, so they focused mostly on sequence similarity and network alignment. It's the new millennia that brought us the first draft of the human genome, as well as a bunch of data to train models on, and that's when we start to see more supervised machine learning experiments. It's not until 2017 that we begin to see deep learning methods begin to appear. And not until 2023 that pre-trained protein language models use explodes for PPI inference. We've previously contributed two methods here, which are underlined. The first of those methods, rapid, demonstrates for the first time how data leakage affects deep learning models for PPI inference and methods for overcoming that. And that was followed by intrepid. That tackled the problem of cross-species PPI inference. And most of these deep learning PPI inference models sort of look like this architecturally. Nothing too complicated. It's a twin neural network. You give a protein sequence to a protein encoder, and that transforms the amino acid sequences to latent representations. Those latent representations of both proteins are then aggregated somehow, and then they get input into a neural network classifier, and that finally outputs an interaction score. This happens to be a figure from Rapid, but it's representative of many, many, many deep learning PPI inference methods that don't use protein language models. Increasingly, however, when I'd speak at conferences or even with my PhD committee, they didn't understand why I wasn't using those pre-trained language models. And they were right to bring it up. Natural language models have been exploding in number, with seemingly every month bringing in larger models. And while it took longer for them to show up, protein language models, or pLMs, which model amino acid sequences instead of natural language like English or French, have been catching up in terms of parameter size. Here we see a bunch of the pLMs that we use specifically in this study. They range from ESM2 to PROT5 to some of the older ones like PROS. They were released in many, many different years over the course of, well, not many, sorry, between 2021 and 2023 in this case. The training data sets are very different. You have different unirefs, but also the addition of some functional annotation data sets like Go, or some structural ones like Scope. And they're trained on primarily the masked language modeling task, but also some various other tasks like protein annotation, contact map prediction, structure similarity. Um, and there's a big difference in the parameter sizes for these protein language models that we're taking a look at today. There's a good reason for the proliferations of pLMs. They are really good at extracting features from amino acid sequences. That makes them an excellent choice for PPI inference. Here we see a figure from the ProTrans paper, which demonstrates that that representations of proteins generated by their PLMs cluster according to structure and lineage. This is a figure that perhaps people have seen. But this is done at the amino acid level, but also at structural and the lineage level, like I mentioned. So a very rich representation for these proteins. And despite all this, I still find myself a bit cautious. I'm a bit reluctant to adopt pre-trained pLMs wholesale. And that comes from a perennial problem for PPI inference, and that's data leakage. See, in 2012, Park and Marcotte described three different ways to split data when training supervised models. Splitting your data is where we try to get rid of that data leakage problem, generally speaking. The first way to split your data, C1, is how data is too commonly split in PPI inference studies. C1 data splits are constructed by randomly choosing edges in the PPI graph and assigning them to training or testing splits. On the right, you'll see that PPI inference models often perform great on these data sets. So, and we observed this exactly, we observed exactly this in the RAPID manuscript. That's the numbers that you're seeing here are from that. A dramatic change, however, happens to the performance of those same models when you label certain proteins as belonging to the train set and the remaining proteins as belonging to the test set. And then you only allow testing edges to contain at most one training protein in the interaction. That's what we see here in this data set that's labeled C2. That's what Park and Marcotte called it. And you can see on the right, the performance drops drastically. Finally, we have the most drastic change when we restrict testing edges to interactions between testing proteins and training edges to interactions between training proteins. Doing this exposes the extent of data leakage played in inflating our evaluation metrics. And the performance goes to near random. So you can see that in the chart on the right. Generalized methods like RAPID, on the other hand, do a much better job at maintaining their performance across these three methods. And you can see that here. So this is another figure from the RAPID paper where you can see on the left that C1 data set where data leakage may be at play here. You see all the methods performing pretty similarly, quite well, except for the one in green, which never really performed well. The RAPID is in black here. In C2, we see that performance kind of holds up. But by C3, we see that the performance degrades significantly across all these models, but that RAPID maintains the majority of its performance. And this is not really, it's not so much to do with the architecture. The architecture of RAPID is pretty simple. You can see it here. It has 3 bidirectional LSTM layers. It gets fed into a 2-layer fully connected classifier. Overall, quite simple. Really, the advantages we've seen come from the use of the judicious application of regularization. And you can see that here with this ablation study where we take apart different contributing regularization techniques in RAPID and see that we get various different changes. For example, a 0.37% point change. Sorry, 3.7% change rather, when you get rid of a regularization technique called average weight decay regularization, and also a 5.4-ish% decrease in performance when you remove the tokenizer. And so really regularization here is what gives us a big uplift in performance when looking at generalized data sets.
Joseph,
Now, if you were, Sorry, yeah.
Yes, I have a question about the perennial problem you discussed of data leakage within the protein-protein interaction. And I'm wondering, is this about the training pipeline, or it's about protein-protein interaction as a phenomenon? Because
Yeah,
if one protein molecule, the training molecule interacts with the testing data set that has been separate, maybe there is a possibility of data leakage. So does that mean, what if the training has been careful and the testing is careful, there's no that leakage? Still, do we anticipate that there will be a data leakage?
yeah, it's a good question. And it's a little bit of both. I think it's both due to the pipeline and also the nature of the data. I think the nature of the data makes it difficult to create good pipelines. So the so people were very careful when they were creating C1 data sets. In their mind, they weren't causing data leakage because they make sure that no interactions, so protein A and protein B forming an interaction, that pair of proteins does not show up in the testing or validation set. So in the minds of the people that created C1, they were having they had a robust pipeline, data pipeline. But it just so happens that the very presence of those proteins in the testing set at all is enough to cause the model to find a trivial solution um, and to memorize and become very brittle.
So the
So
performance drops on a new distribution.
yes, yeah, exactly. So um, at the moment that you have another protein that wasn't in that training set, it really fails to make those predictions. Um, and this is not solely because of C3, as we'll see later. Um, you can have these very good, um, uh, you can have a robust data pipeline that excludes proteins from the testing set that is present in the training. So these C3 data sets um, that still fail on out of distribution proteins. So there's a lot of it's a difficult problem, and this is what partly this is about. This is what all my studies are all about this, and how do we get them to be useful? Um, we haven't found a way yet, in my opinion.
Yeah.
But thanks for the question. Um, so yeah, so if you work on this problem enough, a certain paranoia starts to creep in, like I was discussing just now. Um, and I couldn't help ask the question, do pLMs, do these protein language models, smuggle testing data into the C3 PPI inference evaluation setups? And this is what I mean and how I mean that. pLM-based PPI inference models rely on pre-trained pLMs to encode amino acid sequences for them. So these PPI inference models that use protein language models, they usually use pre-trained ones. And the pre-trained ones, first of all, what we do is we train the PPI inference model downstream of the pre-trained PLM. And we do this on a C3 data set. We make sure that the data set is robust. We make sure that there are no training proteins in the testing set and no testing proteins in the training set. Um, and that way that we minimize the amount of data leakage. The problem is that the pre-trained PLMs that we use in the PPI inference models, they're trained on as many proteins as possible. And that invariably includes proteins that are in the testing set. So it's my hypothesis that you need to get rid of these testing proteins in the upstream PLM because those will leak data. That's my hypothesis that we set to test in this paper. And this is how you test that. First, you need to train a non-strict PLM. So a PLM that is like most PLMs that are pre-trained out there. So if I take Protbert or ESM2 from the internet, this is what I'd get. I'd get this non-strict training set. And then I train another one in parallel that is a strict PLM. And that PLM does not include proteins in the downstream testing set. So it does not include the testing of blue testing proteins in that lower panel. And the way that we do this briefly is that we look for proteins, all proteins similar to testing and validation proteins from the downstream task among the population of proteins the PLM is being trained on. Then, for the strict PLM, you remove those proteins from the training data set. For the non-strict PLM, we make sure they were among them. So that way we ensure that we're really testing whether the presence of these testing proteins in the non-strict data set is what causes the difference between these two data sets. Proteins that weren't in the evaluation set of the downstream task were eliminated from the non-strict PLM data set until both strict and non-strict PLMs have the same number of proteins to train on. There was just one problem. Training PLMs from scratch is really expensive. On the double log graph of GPU memory requirements and forward pass speed, things in the bottom right corner are cheaper to train, and things in the top left are more expensive. Networks like Rapid, my previous method, we're used to training those, and they are far, far cheaper to train. We were going to have to train both a strict and non-strict variants of these PLMs to boot. So that's really, really difficult for us in terms of time and money. So that's why we came up with SqueezeProt. And it's a protein language model which is far cheaper to train in terms of both forward pass speed and memory requirements. SqueezeProt was trained using the SqueezeBert architecture, which uses grouped convolutions for the feed-forward network of the transformer networks. And I really recommend this paper. It's really a fun read and really accessible, but it explains the architecture that was used for the SqueezeProt model that allowed us to get such great speed gains. So now that we've figured out how to train these non-strict and strict PLMs, all that it's left to do is to train a PPI inference model using those pLMs, those protein language models, as frozen encoders and compare testing performance on the downstream PPI task. A difference in performance might suggest that data is leaking. And before I jump into that experiment, I just want to illustrate two things. First, when SqueezeProt is trained on a similar number of proteins as leading protein language models, performance is very comparable, showing that this isn't some straw man model. This is a model that performs very similarly to competing models. The green SqueezeProt U50 marker shows results from SqueezeProt trained on about 52 million proteins from UNIRA 50. Another thing to note is how fast SqueezeProt is. It's about 5 times faster than the fastest model we benchmarked. The orange markers here show SqueezeProt SP. This is the SqueezeProt trained on over 500,000 proteins, so it's much, much smaller. But the result is that it's much quicker to train. So we were able to train it on one RTX 3090 for 7 days, whereas the U50 took a month on 4 RTX 3090s. That's to be compared with the other models, which here were trained on what the authors called the world's second fastest computer. So we don't have a ton of resources in our lab, or we try to use as few as possible. And also we like to make sure that our research is reproducible by people who don't exactly have the world's second fastest computer to run things on. So yeah, so this is a much, much cheaper experiment. It also is so much cheaper to train overall. Oh, sorry, that's what I just said, yeah. But regardless, no more stalling. Here are the final results. The PPI inference models rely on the non-strict PLM. The PPI inference models, which rely on the non-strict PLM, outperform an identical model, where the only change is the strict PLM. When you train using the strict PLM, you have a 12% worse result. than the non-strict one. This was a statistically significant result, and it illustrates that there is data leakage occurring here, because the only difference here is the presence of testing proteins in the training set for the protein language model. That's the only difference between the blue and the red graph. So even when we bring down the maximum similarity threshold between training proteins and testing proteins, so we change how much a protein has to look like another protein before we call them similar, this result still remains the same. So you can see here the blue line and the red line still maintain that difference across this chart, even though the maximum sequence identity changes. And we wanted to find out whether this data leakage was present for all the tasks. So we devised a keyword ranking task. We didn't just want to try it on PPI. So let's call back to the ACE2 protein we saw earlier. We train a model which ranks 1,086 protein keywords from UniProt according to their relevance to the protein. So in this example, it correctly ranks host virus interaction and metalloprotease in the top spots, but incorrectly ranks ATP binding, something that it doesn't do, above cell membrane, which is something it does. So each of these keywords belong to a category. Proteins in the testing set of the keyword task were selected such that they were the same testing proteins as the PPI inference task. When the only thing we swap out is the strict and non-strict pLMs, we really don't see much of a difference in performance except for that of cellular component category. So this sort of indicates that the data leakage really only depends on what task you're evaluating on. And I'd speculate that the pairwise nature of the PPI task is where this data leakage is most relevant. So unless you're doing a pairwise task, the data leakage may not matter that much. But for PPI inference, it seems to be a big problem. The PPI networks that SqueezeProt infers are both realistic and resemble that of leading pLMs. So we just wanted to make sure that the results of our SqueezeProt inferences match those of other models. And what we see here is the degree of proteins in the network inferred by the strict variant of SqueezeProt SP, and that's plotted against the degree in the ground truth network, so the training data set, basically. We see that the Pearson correlation coefficient is really high. So it is constructing graphs that have a similar degree as the training data set. And the degree of the inferred network is also similar to that of other pLMs like ESM2650. And that holds true between SqueezeProt and all the other pLM-based PPI inference methods we've tested. It's not shocking to see that the SqueezeProt SP variants make very similar predictions to other PPI inference methods. On the left, we see agreement as measured by the Cohen's kappa coefficient between the non-strict SqueezeProt SP models with both pLM and non-pLM methods. The blue markers show the same thing for the strict variant. Overall, both strict and non-strict models agree similarly to these methods. We also came up with a measure of concordance between models, which controls for the skill of the model, which we call skill normalized concordance. And the reason why we came up with this was because you can see the difference in performance in existing concordance measures like Cohen's kappa. So here you can see the non-pLMs have a lower Cohen's kappa than the pLM ones. And that's strictly just because the non-pLMs make less accurate predictions. But we wanted to measure agreement while controlling for the differences in ability between the different models. So that's why we came up with this idea of a skill normalized concordance. The details are in the paper of how we came up with the skill normalized concordance. But I think I would love to hear what people here think of this metric, because we came up with it ourselves. And obviously, I think it's cool because I came up with it, but I'd love to hear people's opinions after reading the details in the paper. So now you have regular vanilla attention-based models And these models come with quadratic memory complexity related to the length of your sequence. So it's sort of n squared, where n is the length of your sequence. Truncating at the context length of the pLMs here has some potential implications. This is what people usually do to limit the amount of memory complexity they have to deal with. And the context lengths here for proper is quite high. It's 2048. And only 0.4% of all proteins that we know of in the human genome are truncated at this length. But some of them, like PROT5XL, truncate at 512, where 20%, almost 20% of proteins are truncated. So there are some serious implications there, perhaps, for how this affects performance. Perhaps there's something in that is relevant for performance. Despite this, though, we really didn't observe any reductions in performance due to context length. Here you have ESM2650, and each bin in this plot represents pairs of proteins where the lengths of each protein in the pair is indicated by the two axes. The color of the bin indicates performance. So the redder it is, the better it is, and the bluer it is, the worse. For ESM2650, as is the case with all of the other pLMs we tested, there's not really a trend. The red circles persist in even the proteins that are truncated at quite low lengths. Sorry, let me restate that. That was incorrect. The red circles appear also in long proteins that exceed the context length of the model, which is perhaps unexpected if context length was truncating important parts of the protein sequence needed for PPI inference. So what we're seeing here is most likely this idea that these PPI inference models are taking a very high-level look as to whether two proteins interact and are not really looking fine-grained at a high resolution at these sequences. And you can see this as well. This is another way of seeing this, where the context length here of each model is reported by the dotted line. You have the maximum sequence length on the x-axis of the proteins. So in the pair, which the longest sequence is reported on the x-axis. And the lighter color shows you the proportion of correctly labeled edges in that bin, and the dark colors show the incorrect ones. And as you can see, there's no difference between the strict and non-strict models. And similarly, when you pass that dotted line, it's not like the proportion seems to get much worse. It seems to be mostly the same overall. So really no correlation between the PPI inference length here. Next, we were curious to see how well these pLM-based PPI inference models fare on very out of distribution proteins. So to call back to our initial example, we want to evaluate methods on the network of interactions between SARS-CoV-2 proteins and human proteins. Perhaps there are some differences between these models based on the strict and non-strict SP variants that will appear due to this. You'd expect that the data leakage perhaps would cause the strict model to perform better than the non-strict model. But we're not seeing that here. In fact, we see that the strict and non-strict models basically perform the same. So here we have the non-strict in red and the strict, we have the strict model in red and the strict model in blue and the non-strict model in red. And there is no real pattern here. They seem to mostly overlap. The dotted line is the human test set. The solid line is the SARS-CoV-2. As you can see on SARS-CoV-2, they perform near random. The gray lines are other pLMs. And as you can see, the trend for SARS-CoV-2 is very much random. The other pLMs are not able to, pLM-based methods are not able to do a good job at inferring these things. And this sort of paints a picture, and we'll see this with the next example, the next experiment as well, that these PPI inference models are taking a very high-level approach to inferring these interactions. They are not quite looking at sequences at um, uh, low resolution, at amino acid resolution, it seems. And they are still quite brittle. So C3 is not sufficient to, my hypothesis is that C3 is not sufficient to generalize these models. You need something more to create generalized PPI inference models. We're not there yet. And further indication for this is our final experiment that we're going to be looking at today, which is evaluating whether small but important changes to the amino acid sequence, which affect protein binding, are reflected by the PPI inference models. So more specifically, we first collected changes in Gibbs-free binding energy between wild-type protein pairs and mutant protein pairs, and that all came from the SCEMP2 database. So the idea being that if you have a point mutation in a protein that changes the free binding energy between between that protein, that mutated protein, and the wild-type protein, that that changes the free binding energy, right? So that's the change in delta delta G. We want to measure the difference in inferred interaction score from the PPI inference models between wild type and mutant pairs. So let's say we infer the probability of interacting between the wild type pair and the mutant wild type pair. And we want to see if there was some kind of relationship, some kind of correlation between that change in inferred performance and the change in free binding energy. Does everybody understand that setup? Okay. So when we looked at this relationship, we really saw nothing. There was really no correlation. This is true for all of the pLMs that we studied. And this indicates that the pLM-based PPI inference methods are really not sensitive at all to these small but very important changes in amino acid sequence. So these PPI inference models are still not doing a good job at finding very small changes, very at an amino acid resolution of these models. So there's something that is missing here. So in conclusion, our main findings are that non-strict pLMs inflate performance of PPI models. We outline a pipeline and pre-compute data splits for training strict pLMs, and also tested efficient architectures for training pLMs on commodity hardware. The pLM data leakage depends on the downstream task. Maybe it's only on pairwise tasks, maybe primarily on PPIs. And then finally, pLM-based PPI inference improves performance, but there are still some very open problems. It improves performance, but the generalizability is really, really a sticking point. Mutation sensitivity remains a problem. Virus host PPI prediction remains a problem. So finally, I'd like to thank my lab, my supervisor, Amin Ahmad. Everybody here is shown on the right. And also the funders, the people who helped this happen on, we didn't have the largest budget, but the one that we did came from generous funding from primarily the government and our university. So thanks so much. If you have any comments as well, feel free to reach out by email or on social media.
Thank you, Joseph. Wonderful discussion. Of course, there are a few questions from my end.
Sure.
Members, if any one of you has a question, you just unmute yourself and put it out. Before I go to Gabriel's question, mine is about the intrinsic nature of this discovery. Like, when you say somewhere you've said that you realize that the context length doesn't affect the performance of the pLMs. When you look at maybe the variations in the sequence length. And that is surprising because a lot of noise is being made about context length and context. So any further like sort of highlight why that?
Yeah. No, I think we looked at these these individual things expecting the things that you mentioned, right? We expected to see worse performance on proteins that were longer than the context length. I think it's a rather sad explanation is that I don't think yet that these models are making true inferences on the biology of these proteins yet. I think that it is still some degree of memorization. There is what we're trying to do is we're trying to reduce the brittleness of these models, the memorization of these models, but we're reducing it. We have not at all eliminated it yet. It is still very early days for PPI inference. In terms, even though we've been studying this since '96, or at least we've been studying the deep learning models since, let's say, 2013. There is still so much for us to figure out, because for a long time we were training on C1 and many people were reporting that we were 91% accurate on these tasks. And so people were satisfied and progress wasn't necessarily being made in the most relevant places. So I think, yeah, with context length, I think it's just a symptom of the model memorizing these proteins and being able, from very small sequences, being able to guess at, the identity of the protein and whether it interacts.
Gabriel asked a question, and it sort of like hints on that. He was asking how do embeddings from protein language models compare to handcrafted biochemical features in predicting protein-protein interaction. Mike,
Oh, so I see
like,
it. Sorry, I didn't see the chat. Yeah.
yeah, it's there, but I'm wondering, do you handcraft the biochemical
Uh,
features or like
there's a long history of like, especially before protein language models, of creating these sort of matrices that, and it's a tradition that comes from the success in DNA. So like people may be familiar with the blossom matrices that are useful for DNA. People have tried to come up with this for amino acid sequences, and they come up with something similar, or at least on the basis of something you see in the Prop T5 chart. Here, where you have the hydrophobic, aromatic, aliphatic, positive, negative, polar, neutral. They'll annotate amino acids with these things and sort of create these large vectors by concatenating these vectors with those properties. Historically, these have done quite poorly, frankly. And I believe there have been a couple of studies, I can't name them off the top of my head, but there have been a couple of studies that do directly compare some of these more handcrafted annotations. I don't think they would be terrible as inputs to something that extracts features. But like if you like send them to an SVM or something like that, overall they tend to be do quite poorly. But I think there's room for example, like there's a lot of room in this space, I think, for tokenization. I think if there's if folks have a idea of how to use these handcrafted features to help create some initial tokens. But the thing is that what we're finding is that these models learn these things by themselves. So that's why we see them clustered here. The hydrophobic ones sort of cluster, with the hydrophobic ones and vice versa. So you can do it yourself. You can add it. But there's this, it's always a delicate balance between what's an inductive bias and that you're adding to the model, or what is just making the model worse because it isn't able to learn the best representation for the downstream task. So my guess is that it probably wouldn't be very good. It probably wouldn't increase performance, but I don't think it would. And I think there's maybe a risk that it would hurt it, but on balance, it probably would be similar.
Okay. He's also, I think, extending the
Yes.
conversation, asking about the strategies that can reduce computational overhead.
Yes. Um, so yeah, if you have a large scale PPI inference, PPI network, one thing that helps when applying these pLMs. So you know, the idea being if you're trying to infer PPIs, one thing that helps a lot is the fact that the um, graph is pretty sparse, um, and there's a lot of high degree nodes. And so what you can do is pre-compute these embeddings for the proteins, um, and then use them to infer later interactions. So a protein with a very high degree, let's say it has 500 interactions, because there are some that do, while the median I think is like 20 or 40. Um, that helps reduce your computational overhead by a lot because the comparing the two embeddings usually is very cheap. You know, in many of my models, I just use two fully connected layers. Um, but the pre-computing part is where you use it through the transformer. So usually you just have to embed a protein once, um, and then infer its many interactions pairwise. So that usually increases speed a lot. Um, other computational strategies is to use more lightweight protein language models, like the squeeze prop model that we have here. The reason being, um, the thing is that like our model is, like we said, 5 times quicker and uses much, much, much less memory. That being said, it is less accurate. You know, if you want the best um, protein language model, you're probably going to be using something like ESM3 right now, which, depending on the model usage, is not necessarily, a ginormous model. Prot-5 XL, I think, is bigger, for example. So anyways, using the right pLM and pre-computing your embeddings, usually the way to uh, reduce the computational overhead for PPIs. Actually, what's interesting is one of the big limitations for us has been actually disk space. So we're able to find ways around the GPU memory and find good running times, but we have to compute so many protein embeddings and save them to disk. And we end up with many, 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 many terabytes of data. So disk space is actually what is our bottleneck often.
Wow. Thank you, Joseph. Somewhere you stated about like the non-strict pLMs inflating their performance,
Mm,
and it's, I mean, like, what does that even mean? Like,
yeah.
in sort of
Let's
like
see.
deconstructing it.
Yeah. No,
Yeah.
it's a good question. Let me see if I can, yeah, here. Um, so the idea is that these models have seen overwhelmingly the same data. So the percentage of proteins that are different between strict and non-strict is actually very, very, very small. We're talking about on like 1 or 2%. That said, but even with that change, we get a 12% increase in performance. Um, and the question is this model actually 12% better at this task? Or is it 12%, does it have a 12% advantage on the specific testing set? So this is a question that sometimes comes up in the sense that like sometimes you know exactly what you want to infer. You want to know whether these two proteins are whether a set of target proteins are going to interact. And in those cases, sometimes it's okay to train on the data that you're going to evaluate. But here we're in the business of benchmarking. We're in the business of trying to see, in the general sense, are we performing better? And the idea here is that this setup, by using these pre-trained pLMs, you are having a misconception of how well your model is doing. Not by a ton, but by 12%. So there is a your model's doing better than you think it's doing, not because of your choice of architecture or the choices that you're making, but because of 1% difference in the data set because it's seeing it in the testing set, so it knows the answers to the exam it's writing. Um, sort of like if you take two students and you give them the exact same um, curriculum and you teach them the same course, but you give one of them some of the answers. It's going to perform that second student probably is going to perform a bit better. Um, and you're not actually evaluating whether the two student whether that student when they go on to write another exam where they don't have the answers to it, whether it'll do a good job. Um, and so that's sort of um, what that means here in terms of inflating performance.
Wow. Okay. Thank you very much. The last one
Mm.
is from Badru. He's asking, have you looked at other methods that could do the same thing? If yes, how did they perform? I think he's sounding the other rhetoric of people screaming at you, oh, transformers. Uh,
Yes, yes,
yes.
yes.
Yeah,
Um,
that's the response there.
yeah. So um, it's interesting, let me think about the answer to this. So there are some there's a lot of different ways to think about how to solve this problem. Um, and honestly, it's my favorite part of this. When I started these studies, I thought that I would spend most of my time thinking about interesting architectures, and instead, I mostly think about how to prepare data and make sure that it's the way it's supposed to be. Um, and so it is a lot of fun to think about like the possible solutions. You can I think there's a lot of value in trying to imagine how older models might perform, k-nearest neighbors and SVMs, if they're given the right features. Um, sort of like if thinking of the pLMs as a kernel trick. But let's say pLMs are the problem. Let's say it's the paradigm that's the issue. I don't necessarily think that is the case, but it could be. Um, then it could be that that more parameters and more sophisticated architectures are the answer. Unfortunately, a big problem that we have is just data availability. So the data available to us is not such that we're able to, um, well, the data that the data that in the way that it is currently used is not available to us in a way that we're able to construct large data sets. So for example, um, where is it here? So on Rapid, we um, used as large a data set as we were capable of using, as large a high confidence one. And in the bottom two rows, we replaced the encoder with a transformer, a large one and a small one. And these weren't pre-trained transformers, and this is the difference between the pLMs and this. And what we saw was a 15%, the biggest negative difference, 15% difference and a 6% difference in performance. And that was just due to overfitting because there were so many parameters and so little data. Pre-trained language models get around this by not training on protein-protein interaction data, which is much smaller, but instead just on protein sequences, which is in the mass language model question. So in those cases, you have much, much more data. So that's how pLMs are able to get around this sort of limitation in the number of data points we have. For example, we only have 200,000, let's say, data points in Rapid, I think. And here we're looking at, do I not? Oh, I guess I don't have, yeah, I have training sizes here, but it's being covered um, by Zoom here. Let me see. Yeah, so here we have 66 million, for example, in one of the smallest assets of training data. So that's a lot of training data that we just don't have for Rapid. So the short answer to this is we've looked at other methods. People have been looking at methods since 96. Um, but we've yet to really find something that works better than what we're doing now. But the main issue is that we don't know how well things really are working, right? Because we have this data leakage problem. And so first, in my opinion, what we need to do is make sure that we have our benchmarks correct and then, or useful benchmarks, and then be able to evaluate other methods.
Wow. Thank you, Joseph. I think that brings us to the end of this discussion, unless there are other questions. But I want to thank Joseph for saving off time and really discuss this work with us. We're really honored and excited. And to the members, thank you very much for joining. Has been a good discussion, enlightening, and really appreciate Joseph. Finally, we've had it after a long awaiting.
Apologies for that. You know, I can figure out this stuff, but time zones I still have problems with. So
Exactly.
thank you so much, everybody, for your patience, and the honor is all mine. And thank you for the questions.
All right. So once again, thank you, Joseph. Thank you, everyone. And I want to say goodbye to you, Joseph, in particular, and to everyone who has been here.
Cheers. Thank you so much.
All right. So goodbye, everyone. See you in our subsequent talks, perhaps next week.
Take care.
Take care.